This is the chemistry of life. Here is the periodic table. On the periodic table, there are certain things that we'll need to know. The 6 most common elements of life are listed below. We have sulfur, phosphorus, oxygen, nitrogen, carbon, hydrogen. These are otherwise known as sponge. When looking at the periodic table, there's a couple of things that we need to know. First, the letter is the symbol name. Above the letter is the atomic number. For carbon, the atomic number is 6. The atomic number is equal to the number of protons. In a stable atom, which is fairly rare, the number of protons is equal to the number of neutrons, which is equal to the number of electrons. The number below the symbol name is the atomic mass. When looking at the periodic table, one thing that we need to know for each element is how many valence electrons are available. If you remember, valence electrons are the electrons that form bonds. There's an easy way to tell how many valence electrons there are. If you look at the group number just above the column, here we have 13, 14, 15, 16, 17. The second number, the 3, tells us how many valence electrons. Boron has 3 valence electrons. Carbon has 4, nitrogen has 5, oxygen has 6, fluorine has 7. Anything in that column is going to have the same number of valence electrons. And if you remember, the element needs to have a full octet. So that will determine how many molecules or how many other atoms can bond with that atom. Carbon has 4 valence electrons. To fill up its valence shell, it would need 4 electrons to make that octet. Thus, carbon can form 4 bonds. Looking at the different types of bonds that we'll need to know, helium has an atomic number of 2, meaning it has 2 protons, and then in a stable atom, it would have 2 electrons and 2 neutrons as well. The protons and neutrons are within the nucleus of that atom, and the electrons float in the electron cloud around that nucleus. The diagram that's a little bit more accurate to draw would be the orbital shape diagram here. The electrons tend to float around the nucleus in specific patterns. In carbon here, you can see these balloon-shaped orbitals that they float around. And remember, it is a 3D object, so they're going to project in all 3 dimensions. The 2 major types of bonds are covalent and ionic. Covalent bonds are bonds that share electrons between atoms. Carbon really likes to form covalent bonds. Because it likes to form covalent bonds, it gives variety to the shapes of molecules that it can form. Ionic bonds are a transfer of electrons. So you'll have a positive ion and a negative ion, one pulling that electron further away from the other atom, giving positivity to one side and negativity to the other. Ionic bonds tend to happen in our salts. Some weaker bonds, hydrogen bonds, are found between hydrogen and other highly electronegative atoms. Water forms hydrogen bonds. The bond between the hydrogen and the oxygen is a weak bond. It's weaker than a covalent bond. Hydrogen bonds give water some pretty cool properties that we will talk about later. Van der Waals forces are close attractions between molecules. In the example of the gecko's foot, the molecules within the foot have a close attraction to a molecule on the wall, and it's a very weak bond, but it's very easily made and very easily broken, and it allows the lizard to walk on upright surfaces. Shape is very important to molecules. Shape is going to determine our function, and the bond type and the way that the bonds form will determine the shape. There are isomers. Isomers are having the same molecular formula. such as C6H12O6. Their glucose and galactose both are C6H12O6, but the way that the molecule comes together is a different shape, thus giving it a different function. There are a couple different types of isomers. We have structural isomers, which are what I was just talking about, where it has the same molecular formula, but the way that the molecule comes together is a little bit different. A cis-trans isomer has a kind of a back and forth. So one molecule is kind of thumbs up on both, and the other molecule is thumbs up and thumbs down. So it's a kind of a twisting within the molecule that causes that. And because those molecules are not on the same plane, they're on different planes, gives that molecule a different shape, thus giving it a different function, and it behaves chemically different. And the last is enantiomer. Enantiomers are the left and the right. So one molecule is kind of the left-hand version, and the other molecule is the right-hand version. They look very similar, but they are slightly different. One of the biggest places that we've seen problems with enantiomers is in a drug called thalidomide. Thalidomide in the early 50s, 60s was used in pregnant women for morning sickness. Thalidomide completely eliminates that nauseous feeling, so a lot of pregnant ladies were taking it to get rid of that morning sickness. But there was a great drawback. One version of thalidomide was perfectly safe. The enantiomer is the molecule that causes problems, and thalidomide easily goes back and forth between these two, and there's no way to keep it from going to the dangerous version of thalidomide. The thalidomide was causing birth defects, basically eliminating arms and legs, and the hands and the feet were growing directly out of the shoulders and the hips. Thalidomide is still used for some cancer treatments, but is not used in pregnant women anymore because of these severe birth defects. Isotopes. Isotopes are atoms with a different number of neutrons. Here we have a picture of hydrogen isotopes. Hydrogen 1 is that there's one proton, so it's missing a neutron. Hydrogen 2 has a proton and a neutron, so that 2 is telling you how many protons and neutrons there are. And hydrogen 3 is 2 neutrons and 1 proton. One isotope that's used commonly in biology is carbon-14. Carbon-14 is used for radioactive dating of fossils. Carbon-14 has 6 protons, and so 14 minus 6 is 8, thus meaning that the carbon-14 has 8 neutrons. Radioactive isotopes have a nucleus that is not stable, and it breaks down over time, and it tends to break down at a very even pace over time. That is the reason why we can use carbon-14 in order to date fossils. We know how long it takes for carbon-14 to break down, and we can look at how much carbon-14 is broken down over time, giving us a date range of that fossil that carbon-14 is within. We can also use radioactive isotopes as molecular markers. Some radioactive isotopes have a short half-life, and they can leave the body fairly quickly. And as the radioactive isotope is within the body, it emits particles, and a CAT scan or a MRI can pick up the movement within those radioactive isotopes, kind of causing a glow within the picture. It can also be used as a marker, where you can inject the isotope and it can attach to certain molecules, and you can measure the amount of that molecule in a particular spot over time, trying to trace where that molecule is going through the body.